All right, so it's 10 o'clock local time in Colombia now. Welcome everyone to the Ensemble tutorial. I hope you're doing well. My name is Luis and I am joined here today by George and Juan, who will be assisting answering questions in the background. So let's just introduce ourselves. So I'm Luis, I'm part of the Ensemble outreach team, and our team is in charge of the Ensemble help desk. And we also provide workshops and courses across the world like this one. Yeah, off to you, George.
Hello, everyone. My name is George. I'm the Ensemble Outreach Project lead. So I'll be here today to give you some support on the live document and answer your questions in the chat. And it's very nice to meet you all. Next up, Juan.
Hi, good morning, everyone. My name is Juan Pablo Narvaez Gomez. I am an Arise Fellow in the Ensemble team at Ensembl EBI, and I work with the development of infrastructure to integrate genomic and biodiversity data. Today, I will be in the background helping to answer any questions you may have about Ensemble Beta. So thank you very much for joining us today.
Thank you both. Yeah, I should have probably mentioned that I will be presenting. So yeah, if you have any questions at all, please don't be shy and ask them away. There's plenty of help in the background here, as you can see with George and Juan. And I'm very excited to say as well that we have a fellow Colombian here. So Juan is from Colombia, so this is great. Okay, so let's begin, if my slides allow me. Okay, so let's just start with a bit of background. So we work for the institute called European Molecular Biology Laboratory, or EMBO. And this is an intergovernmental organization, and we have 6 sites across Europe. And each site focuses on a particular field in biology. So our main lab and administrative center are in Heidelberg in Germany. So that's the main headquarters of Ensembl. We have structural biology labs in Hamburg and in Germany and in Grenoble, and epigenetics and neuroscience in Rome, and bioinformatics in Hingston in the UK. And Barcelona is the newest site, and that institute focuses on tissue biology and disease modeling. So George, Juan, and myself are based at the European Bioinformatics Institute, or EBI, and it is the second largest EMBL site in Europe. We have about 800 employees at the moment working at EBI. And what we do is we provide free bioinformatics resources. We also have research groups focusing on computational biology, and we provide bioinformatics training. So Ensembl EBI offers a number of different resources, some of which you may have come across before. We've got the European Nucleotide Archive, or ENA, where you can deposit sequences and make them publicly available. We also have the worm-based database, where you can find any parasite information and genomes. And we've got Ensembl, the Genome Browser, which is probably one of our most popular and most used EBI resources. So for this workshop, or tutorial, I hope to show you the new Ensembl browser. So we've developed, well, our developers have developed a new browser, a much faster one, and we're going to walk you through how to navigate the new website today. I'm going to show you how to find what type of data that is currently available in the beta site. We're going to explore the different species that are available. And I'm going to show you, of course, where you can find help and documentation once you're out there on your own. So the tutorial will follow this structure. So I will begin with a presentation where I will introduce you to the data or the tool and how it was processed. And then I'll move on to a live demonstration where I will walk you through my own screen, how to find this data on the Ensembl browser. And then you'll have time for exercises. So this is where you try exploring the browser out yourself. Just remember that this is not a test, so please don't put any pressure on yourself. If you have any questions at all, please feel free to enter them into the Q&A in the chat on the platform, or you can also enter them into the living document, which I will show you now. All of the training materials are available on our training site, which is training.ensembl.org. And I'm going to show you how you can access the site on my screen now. So I'm going to switch to my browser, and I'm going to type in training.ensembl.org, ensemble without the E at the end. And let me zoom in a little bit so you can see it a bit clearer. At the top of the page, there is a link that says training materials. So I'm going to click on that. And our training page is the second link here. So the one that says ISCV LATAM. And I will also share the direct link to the training page. There you go. George already shared the link so that you have it handy. You won't have to look for it anymore later on. So this is our training page. Any training materials you will find in here. So on the right-hand side, we've got a link to the conference website, which includes the agenda for the entire conference. We've got a link to the presentation slides. So these are the slides that I will be presenting today. And we also have a link to the living document, which I mentioned just now in my presentation. If you open this living document, this is just a Google Doc with some information about the course, the agenda, some helpful links, and most importantly, on page 4, there is space for questions. So if you have any long questions, or if there are any screenshots that you'd like to share, I suggest you use the living document because you'll have access to this indefinitely. So after the workshop, we will keep this document open. So if you had a question and you've forgotten the answer to it, you can always come back to this document and find out the answer. But alternatively, you can also enter your questions into the Q&A on the Juno platform as well. So I'm gonna go back to the training page. I suggest you leave the living document open for the rest of the tutorial. So back on the training page, if you look underneath the living document, there is also a link to a Slido poll. So we'll be using a few online polls today. So I suggest you open that in another tab as well. There will be a lot of tabs today. And you can just access the same link for all of the questions. So the link is not going to change throughout the tutorial. Scrolling down, we've got a few subjects. So these are all subjects I'm going to cover during the tutorial, except this very first one, because this very first one focuses on the old or the current Ensembl browser. So I've simply added this in case you're not familiar with Ensembl. You can open this and view some screenshots on how to access the different data on the current side. But for our workshop, we are going to focus on the new side, on the Ensembl beta side. So we've got, again, a few screenshots of the demonstration. This is what I'll be going through in my live demonstration on my screen. And under exploring gene builds in Ensembl beta, you can find the exercises that we're going to go through in the practical session later on. Okay, so I will leave this open as well, and I suggest you do too. Okay, so off to our first poll. Please join me at slido.com. You can enter this code into the website, or you can use your phone and just scan this QR code, and you should be ready to go. I'll give you a couple of moments to join. Okay, oops, sorry. All right, so I have no questions just yet, so it might be blank for you, but please leave that page open because there's gonna be a few questions coming up in a minute. So our agenda for today, I'm gonna first start off with a brief introduction to the Ensembl project and the new Ensembl browser. We're then gonna move on for a short break or a medium short break. Please feel free to leave your computer and go get yourself a coffee and stretch your legs. Then we're going to continue on at 12 with the practical. I'll also be talking about how we annotate genes and transcripts in Ensembl. And then at the end of the workshop, there'll be time for some questions, feedback, and I'll wrap everything up with a few finishing slides. Okay, are there any questions in the meantime? I might just share the poll, the link to the poll as well. Sorry, I'm just trying to find it. Here we go. Okay, no questions so far. All right, yeah, we haven't really covered much yet, so there we go. So just a brief introduction to Ensembl and the project. So in 1977, Fred Sanger and his team sequenced the very first genome, which was a bacteriophage. It was about 5 kb long. And this really kickstarted the era of sequencing and sequencing much more complex organisms. The first draft of the human genome was published in 2004. So this was a project that started back in the 1990s. And since then, many, many more organisms have been sequenced. And today we can sequence an entire human genome in a matter of days. So we've come a very long way since that very first draft of the human genome. The Ensembl project was launched in 1999, and the aim back then was to annotate this draft of the human genome and integrate it with other databases and resources and make it publicly available through a platform which we call now a genome browser. So out of curiosity, please go to the Slido poll and enter what species you're currently studying or you have studied in the past. And I'm curious to see if we have that species available in Ensembl. You can enter multiple species as well if you like. So I'll give you a few moments for this. So far human, well, congratulations, Ensembl has loads of different human genomes. If you are studying any other genomes, I will show you during the demonstration how you can find out what species are available in Ensembl. Okay, so as we expand our knowledge in genomics and additional data being constantly published, a new version of Ensembl is released about every 3 to 4 months. So for each release, what we do is we add any new genome assemblies, we update any underlying software, we update or add any gene sets or gene annotation data, and we perform additional comparative genomics analyses. And because Ensembl is not static, you want to make sure that you write down what Ensembl release you're working with. So the current release is Ensembl 113, which was published back in October 24. And the next release, 114, we're expecting to come out in February 2025. So, while the current Ensembl website follows this release pattern, the new website will also follow a similar pattern, but we won't be sure how often it will be updated just yet. But just know that it will work as a release cycle, and it will constantly be updated by the Ensembl team. So why are genome browsers important? Sequencing, as you might have guessed, has become much cheaper and faster. And because of that, genomic data is piling up very quickly. But when sequencing data is produced, you get something like this. So just a bunch of A's, T's, C's, and G's. And if you try to read this sequence without any annotation data, it wouldn't really mean anything to you. It would be like trying to read a new language without a dictionary. So this is what genome browsers are there for. Genome browsers map annotations and different genomic features to a genomic region, giving that region value. And they also allow you to display genetic data in a visual way. They help you explore and interpret the data and find out what neighboring genomic features you can find in that region. And what Ensembl does specifically is it brings together information from a wide range of other databases into a single site. One thing I want to emphasize, though, is that Ensembl does not do its own sequencing. We simply take publicly available sequencing data, and we use our own annotation pipelines to annotate the sequence. We have many features that we annotate in Ensembl. So these include genes, variants, promoters, binding sites, and many, many more. We've got variation data in a number of different species. We offer pre-computed comparative genomics data, so alignments, gene trees, and homologs. We also have regulatory builds for human, mouse, and a number of other popular species like rat, pig, and cow. And Ensembl also offers a number of different tools that you can use to analyze your own data. So for example, we've got the Ensembl Variant Effect Predictor, or Ensembl VEP. And this tool allows you to annotate your own variants and find out what the consequences are of these variants. We also have the Biomart tool, which lets you export large amounts of Ensembl data without needing to know any programming language. So there is a nice little user interface that you can use, and you just need to use your mouse to click and drag, click and select any data that you want to download. You can also access Ensembl data via our APIs and MySQL servers. And most importantly, Ensembl data is completely open source. So that means that you can access all of the data completely free of charge, and you can use it in your research as you wish. All of the data you can access on our FTP site, and the source code you can access through our GitHub repo. Whenever you see anything underlined green on my slides, this means that these are clickable links, so they will lead you straight to those websites. All right, any questions so far? Brilliant. So what's the mission of Ensembl? So we're always aiming to improve the Ensembl resource and make the user experience more, what's the word? More worthwhile and easier to access. So we are a resource to support research in comparative genomics, in evolution, sequence variation, and transcriptional regulation. And since the launch of the Ensembl websites, we've added a lot of different organisms in the browser to understand the genomics behind them. So far, we've got, I think it was about 3,000 vertebrate species and 50,000 non-vertebrate. No, wait, hang on. I'm not sure about the number of vertebrate species, but it's definitely over 50,000. Sorry, it's over 50,000 non-vertebrate species. For vertebrate species, I think it's about 500 or so. I'll need to double check that word, but that number. But basically, what I'm trying to show you here is that since that very first genome that the Ensembl project started with, we've added many more other organisms to the database to support researchers in other communities. So today, if you are familiar with the current Ensembl sites, you might notice, or you might have noticed, that there are several different sites that you need to access depending on what type of species you want to you are studying. So we've got the Ensembl main site, which is dedicated to vertebrate species. So these would include all the human genomes, mouse, rat, and so on. Then we've got the Ensembl Genomes website, and this is dedicated to any non-vertebrate species, and it's divided into different divisions. So we have a separate portal for plants, one for invertebrate metazoa, protists, and so on. We also have a portal for the older GRCH37 human genome assembly. And since 2020, we also have a portal for the coronavirus genome. So since there are a lot of different sites in the current Ensembl infrastructure, it is difficult to make comparisons between genomes of different portals. So this is one of the reasons why we have created the new Ensembl website, is to keep all of the species into a single site. So you won't have to navigate to these different subportals anymore in future. So, and you can imagine that the massive amount of data in Ensembl has a great impact on the performance of the browser. And on top of that, like I said, you need to navigate to the different Ensembl sites depending on what genome assembly you're interested in. And that makes comparisons very difficult and research very difficult. But please know that we are aware of the performance issues, and we are working on a solution to this, and to make the database future-proof, because genomic data is just going to increase even more in future. So in the past few years, our Ensembl developers have been working on very tirelessly on the new Ensembl site, which we currently know as Ensembl Beta. And it's different to the current site with a more modern look and a much better performance. And I hope that during the demonstrations, you'll see what I mean with the new browser being much faster than the current one. So this is just a screenshot of the new website. It looks very different, but it is very intuitive once you know what these different buttons do. So the Ensembl Beta is a new platform, and how it works and how it differs to the current Ensembl site is it's app-based. So instead of navigating the different tabs that you would in the current site, we actually now have different apps that you can access. The infrastructure is completely different as well. So the new site has been built from scratch, and because the infrastructure does not follow the old site, it is now much faster as well, and it's more future-proof. We won't be having as much problem in future anymore when adding additional data to the Ensembl browser, because the infrastructure will be more powerful to deal with that data. It's also more responsive. So in the current Ensembl site, if you zoom into a region in the genome browser, you might notice that the loading takes quite some time. In the new browser, this is much faster compared to the current site. So the zoom is immediate. You don't have to reload the whole page anymore. And it also has a much more modern user interface. So I think we can all agree that the current site has a very 90s look, which is nice, but a more modern interface does help a lot with the user experience. And the new site also has the ability, also gives you the ability to browse multiple species at once. In the current site, you can only browse data of a single species, whereas with a new site, you can select multiple species and explore data in all of them at the same time. And as mentioned, all of the Ensembl, all of the species available on Ensembl will be available in a single platform. You won't have to navigate any of the sub-platforms anymore. But please remember that Ensembl Beta is still under beta testing. So this tutorial is here for our users to give us feedback on the browser. We want to hear your thoughts, and we want to hear what you think about the browser, or if you have any comments on the browser, or anything that you'd like to be added to the browser. So remember that Ensembl is a community project, and we need that feedback from you researchers to make the platform more efficient. So there are three, we're aiming to release the new Ensembl browser in 3 development phases. So the very first version was delivered in 22, a little less, just 2 years ago. And this version had the genome browser available to it. You were able to find more information about certain genomic features like genes and transcripts. And the BLAST tool was available in that version as well. So we're hoping to release the minimal viable product, MVP, in a few months' time. And this will have the same functionality as the first phase, but you will now, with the MVP, you will also be able to visualize variants. You will be able to explore any homologs of a gene, and more genomes will be added to the rapid release, to the MVP. This includes all of the genomes that are available in the Ensembl Rapid Release website. And then finally, the third phase is for the new website to reach the full functionality of the current site. So that means you'll be able to do any vet jobs on the browser. You will have all of the genomes that are also available in the current site on the new browser. And the current browser, which will be the old site, will be retired. Don't worry about that too much yet, but it will be retired eventually. But that process is gradual, so it's not going to come from one day to the other. It will be gradual, and you will have time to get used to the new browser as well. So I mentioned that the new Ensembl browser has an app-based approach to the functionality. So you can explore any assembly and sequence information in the species selector. On the new site, you'll also be able to select multiple species at once and explore them across all the different apps. The genome browser lets you visualize genomic regions, map annotations via tracks, and explore genomic features in their genetic context. So for example, what other genomic features can I find in the neighboring regions? And if you want to learn more about a specific feature, let's say a transcript, you can switch to the entity viewer to explore the gene function, explore any gene relationships, view any any entries in external databases, and so on. Currently, the only tool that is available in Ensembl Beta is BLAST, so you can do alignments against multiple species here.
Sorry, just to check in. I think you're muted.
Sorry, how long have I been muted for?
No, just about 20 seconds. I also thought like I've lost sound, but it looks like you were muted. Sorry about that.
No, sorry, guys. Let me switch back. Do you
Cool.
remember when I got muted?
I heard you speak about plant genomics.
Was it this one?
No, that was way far back. No,
Oh,
it's
oh.
closer. No.
I'll go back to this one here.
Okay, cool.
Sorry, guys. I'm not sure what happened there. We did get warned about a few glitches in the platform, so there we go. Okay, so just a brief overview of what the Ensembl Beta site is. So the new browser has an app-based approach to the functionality. So in the current website, what you're doing is you're accessing different tabs to view genomic data. But in the new site, it's different apps that you're going to access. So the Genome Browser is a separate app, and then if you want to look at more detailed information about a gene, for example, that's a separate app as well. The infrastructure behind Ensembl Beta is completely new as well, so it's been built from scratch to make sure that the database is able to handle the massive amounts of genomic data that becomes available all the time. It's also highly responsive and has a more modern user interface. So the main difference here is if you visualize a genomic region in the current Ensembl site, when you zoom into a region, it refreshes the whole page. But in the new site, it will only refresh that particular panel. So the loading time will be much faster in this case. And the more modern user interface hopefully will be more intuitive for you as well. Users will also have the ability to browse multiple species at once. So in the current Ensembl site, you can only view data of a single species at a time, whereas in Ensembl Beta, you can select multiple species and explore them simultaneously. And I mentioned that the new site will have all of the genomes in a single platform. So you won't have to access those different subportals anymore if you want to look at data in, for example, a bacterial species. So please remember that the new Ensembl site is under beta testing, so it's not the final product just yet. And we do hope for your feedback and your thoughts about the new website. Ensembl is a community project. It is there to support you researchers in your research. And we want to know what's important for you guys in a genome browser to make it more usable and efficient for you to use in your research. Ensembl, the new Ensembl site is released in 3 development phases. So the beta version of the website, the very first version, was published back in 2022. And this had very limited functionalities, so you could browse very few genomes and visualize the regions. We had only a few key genes available in there, And the BLAST tool was also available with limited functionality. The minimal viable product, or MVP, is the more advanced Ensembl Beta version. And what it is or will be, which will come out in a couple of weeks, it will have the same functionality as the Ensembl Rapid Release website. So it will have all of the different genomes that are currently available in Rapid Release. It will have it in that Ensembl Beta phase. You'll also be able to visualize variants, explore any homologies, so gene relationships, and at the same time, the Rapid Release website will be, the support for the Rapid Release website will go down. And then for the final phase, what we're hoping Ensembl Beta, well, it won't be beta anymore. What the new Ensembl site will be is it will have the full functionality of the current site. So that means that you'll have all of the different tools that are also available in the current site that you can use in Ensembl Beta or the new site. You'll have all of the genomes in the new site that are currently available across the different subportals. And the current site will gradually be archived. So don't worry, this is not going to happen from today to tomorrow. This change is gradual, and there will be plenty of time for you to get used to the new Ensembl website, Ensembl Beta. So I mentioned that the new Ensembl browser has an app-based approach to the functionality. You can explore any assembly and sequence information using the species selector. You can also select multiple species and explore them across the different apps at once. The genome browser allows you to visualize genomic regions, map annotations via tracks, and also explore genomic features in their genetic context. So for example, by looking at any neighboring genetic features. And if you want to learn more about a particular gene, for example, or a transcript, you can switch to the entity viewer where you can explore any gene functions, homologies, and find entries in external databases. Oops, sorry. With regards to the available tools, you can already perform BLAST queries in Ensembl Beta. So the power of the BLAST query via the new site is that you can now BLAST across different divisions. So in the current site, you could only perform BLAST searches within that website, so within vertebrate species, within plant species, and so on. Whereas in the new site, you'll be able to BLAST a human sequence against a bacterial sequence, for example, if that's what you wanted to do. If you need help in the new site, there are different options. So you can open the general help and documentation pages. So this includes any guides and more detailed information about how the data was generated. For example, how the annotation was done, how the variants were imported, and so on. You can access data or tool-specific help pages, so whenever you see an icon like this, you'll be able to click it and open a pop-up window that describes the data that you're currently seeing. And whenever you see a very small question mark like this, you can click on these and find a description of the term that you've just clicked on. So for example, what is a TF binding site, which we will actually go through later. And if you can't find help, you can always contact us via the contact form. So this little icon here will be your very best friend. If you click on it, it will open up a form where you can enter your questions. You can now add screenshots as well. And that will go directly to the Ensembl help desk, which George, myself, and a colleague of ours is in charge of. So you'll always get an answer from a real person and not from AI. So the available human genome assemblies in Ensembl Beta are 99. So we have the GRCH37 assembly, the older human genome assembly. We've got the most supported GRCH38 assembly. So this is the one that is most up to date currently. We also have the more recent T2T assembly and gene annotation available for this as well. And we also have genomes available from the Human Pangenome Reference Consortium. So this includes genomes from 48 individuals, and we've got each of their haplotypes. So in total, 96 genome assemblies. And as the name suggests, the Ensembl browser is still under beta testing and therefore still in development. And our colleagues are working very hard on making the features and the data available in the new site. And some of the features that you can expect to come out next are additional genome assemblies. So all of the genomes that are available in rapid release will become available in Ensembl Beta. Gene expression data imported from Expression Atlas, gene ontology or functions and pathways, any associated phenotypes and their publications, additional variation data. While we have variation data already available, this is still limited, and we are constantly adding more variation data to Ensembl Beta. You will also be able to use the Ensembl variant effect predictor in the new site. And regulatory data is being added constantly. And for any cross-references, there are some cross-references already available, so entries in other databases, but this is not complete yet, and just know that this is being gradually added as we speak. Okay, so this brings me to the first demonstration, where I will give you an overview of the Ensembl Genome Browser. But before I move on, let me just check if there are any questions at all. Oh, there's been a few questions already, and George has answered them. That's great. Thank you very much. Okay, no questions so far. All right, so in that case, I'm going to move to my browser and start our demonstration. I'm going to open a new tab and go to the Ensembl Beta site. So to go there, just type in beta.ensembl.org. I will share that link with you as well in the chat. Oh no, that's not the link. Sorry. Here you go. beta.ensembl.org and hit enter. So this is the new, exciting, modern website. It is very different to the current site, but don't worry, we will walk you through on how you can use it most efficiently. So on the homepage, you can find the different apps that I've mentioned. So we've got the species selector, the genome browser, which allows you to visualize genomic regions, and the entity viewer, which will give you more detailed information about a specific genetic feature. On the top, the same links, but just as shortcuts. We also have a shortcut to the BLAST tool to perform a BLAST query, and a shortcut to our help and documentation. So this page gives you any guides on how the data was processed and how you can use the data. On the right-hand side, more help about how to use Ensembl, or the current Ensembl site, and a link to our contact form. So if you can't find any information about something in Ensembl Beta, you can always message us directly using this contact form here. Okay, so how do you use the browser? The first thing you need to do is actually select a species. So we're going to click on the species selector. And you can either search for a species using this text box, or you can select a species from the list down here. And don't worry if you don't recognize the icon. You can always hover over the icon with your mouse, and that will give you the species name. So this one is zebrafish. So let's just explore the different human genome assemblies that are available. So we can see for human, we've got 99 genome assemblies. And if you click on that icon, let me zoom in a little bit. If you click on that icon, you get a list of all of the different human genome assemblies that are currently available in beta. So plenty of HPRC genomes. We've got the GRCH38 genome, so the one that is most up to date. We've got the GRCH37, the older human genome assembly, and the most recent T2T assembly as well. For each of the genomes or of the assemblies, you've got the name of the species, you've got the assembly name or assembly ID, and the accession ID. So this is the ID of the sequence that was originally deposited into an archive. And if you click on that link, it will send you straight to the entry in that archive. So in this case, the GRCH38 assembly was deposited in the European Nucleotide Archive, ENA, and you can find more information about the sequencing, about the sequence itself in here. All right, let's close this tab and go back to the species selector. So the nice thing about the new Ensembl site is you can select multiple species in one go. So for example, if you wanted to explore all of the 3 human assemblies, you can just select them in the table and click Add at the top. And if you focus at the top of the page, you'll notice that these are the species that you've just selected. So as you add species to your species list, this little area will get populated. If you want to find more information about the assembly, you can click on the particular assembly. This will open the assembly information page. So this page contains any information about how many genes there are, when the gene annotation was last updated, where the gene is deposited, so a link to the, sorry, not gene, the genome was deposited, so a link to the archive where the sequence was released. We have any annotation information, so how was the annotation done for the genome. And you can also download whole genome files here. This feature may not currently be available, but please feel free to try it out during the exercise yourself. Okay, if you want to close this panel, just click on the close icon in the top left, and that will send you back to the species selector. Okay, so I've shown you how you can select species. Let's actually look for a gene. So once you've selected your species in the species selector, you need to go to find a gene and enter your gene of interest. So let's just look for BRCA2 in our human genomes. Okay, so once you click go, you get this little list of results, and we've got BRCA2 in all of the genomes, so GRCH38, 37, and the T2T genome. I'm just going to focus on the 38 for now. So let's open the BRCA2 entry for the 38 assembly. You can click on the gene name, and you get this pop-up window that gives you the option to either view it in the genome browser, or view it in the entity viewer, so more detailed genomic information. Let's just visualize it for now. Okay, so I've opened the BRCA2 gene in the genome browser, and this is the genome browser. At the top of the page, you can see it's also indicated that you're currently in the genome browser. And it's, if you're already familiar with Ensembl, it is quite different to the current site. So the main difference being that it is much more responsive. So for example, if I just do the first thing here, if you use your mouse to zoom in and out, you don't get that loading icon anymore. It doesn't try to reload the whole page. Instead, it will just load your region that you've just zoomed into. We still have these tracks. So if you hover over the letters, it will tell you what each of them mean. So G is a genomic track. Oh, if you scroll down, we've got a regulatory track as well. And scrolling down to the very bottom, I'm not sure if you can see that, but there is a variant track at the very bottom as well. On the left-hand side, on the right-hand side, this is what we call the track panel. And here you can turn on and turn off any tracks you might be interested in. So we are currently focusing on the BRCA2 gene, which is this one here. If you want to view the individual transcripts, you can click on the plus number, and this will visualize all of the transcripts. If you look very closely as well in the background, you can see in which direction, on which strand this gene is located on. So it's on the forward strand. Or you can always just click on the individual feature to find more information about that feature. So still quite similar to the current site. You can, the view is interactive and you can click the individual features to open more detailed information. Back to the track panel on the right, we've got the BRCA2 gene here. We always highlight the main select gene, which I will talk more about after our break. And you can expand the different transcripts here as well. So anything that is dark, that has a dark square, like this one here, these are protein coding. And anything that is light or light gray like this one, these are non-coding transcripts, sorry. If you want to find out more information about the transcript, you can always click on this, on these three dots. That will open another panel where you can get the coordinates of the transcript. You can download the sequence. You can find entries in UniProt. And you can also view the feature in the entity viewer. Back to the track panel. Let's close the other transcripts to make it a bit clearer. Scrolling down, there are a few tracks that you can turn off. So at the moment, we've got the GC percentage. You can turn this off. Conservation scores, constraint elements. So you can turn all of these off to make it a bit easier on yourself to view the genomic data that you're actually interested in. So I've just turned off all of these tracks, and now I should only be able to see genes, which indeed, so this is our gene of interest. These are some neighboring genes on the forward strand. These are some neighboring genes on the reverse strand. And then we've got our regulatory track and our variation track. On this page, you can also search for particular coordinates if you like. So while the function is not yet available in the species selector, you can search for your coordinates in the genome browser. So you can click on this navigate browser image icon on the right. And if you want to go to a new location, just select your your chromosome, enter the start coordinate, end coordinate, and click go. And that will refresh this page to the coordinates that you just searched for. Okay, so let's just look at the entity viewer. So let's click on this very first transcript, the main transcript of BRCA2. Oh, one thing I forgot to mention, whenever you search for a particular gene, it will always be, you can always find it at the top of the genome browser. So we will always highlight it as the kind of first track of the genome browser. So this track here only refers to BRCA2. Okay, so I've clicked on the first transcript, the main select transcript, as you can see in this pop-up. You can download the sequence here, if you like, in this pop-up as well. But what I want to do is go to the entity viewer. So if you look to the top of the page again, we can see that we're now in the entity viewer app. And this entity viewer just gives you more information about that genetic feature. So in this case, the BRCA2 gene. This is the gene name. We've got the Ensembl ID, what type of gene this is. So it's a protein coding gene located on the forward strand, and these are the coordinates. In the view on the left, we have our transcripts. The top one is always the main select or the Ensembl canonical transcript, followed by all the other alternative transcripts. If you click on the individual transcripts, it will open this, this menu for more information. So if we close that and open the next one, we can see this is protein coding as well. Maybe go down a little bit and see what type that is. This is retained intron and so on. So for each of the transcripts, you can download the sequence, just like before. If you want to explore the gene function, you can switch to the gene function tab. In this case, we currently have protein information available. So because this is a protein coding gene. Oh, sorry. So if we open the first transcript, the main select transcript, we have predictions from Panther and PFAM, sorry, protein domain predictions from Panther and PFAM. We've got links to some external databases. So if you wanted to find out what function this gene has, while it's not currently available in Ensembl Beta, you can always refer to any of the external databases, which will have that information available. But in the final version of the new site, you will be able to see the gene function within Ensembl. You won't have to navigate to any external databases. You can also explore gene relationships. So this includes homology information. So this is a table of all of the different species and their homologs in them. We have the corresponding gene ID here on the right, and the gene symbol for each of the homologs. And just like in the genome browser, we have this track panel here on the far right, where you can search for a gene, you can download the data, you can explore any publications.
No worries.
Sorry about that. I just got logged out for some reason, so hopefully I won't get that anymore. But we are towards the end of our demonstration anyway. So just the final thing I wanted to highlight here is the track panel here on the left, on the right-hand side. So if you want information about any publications, in this case for the BRCA2 gene, you can view associated publications in Europe PMC, or you can find entries to external databases in, for example, Expression Atlas, UniProt, and so on for each of the transcripts. Okay, so this was just a brief, well, not really brief, but an overview of what you can currently do in Ensembl. Let me switch back to my browser. And we're going to move on with our first set of exercises. And how this is going to work is we're going to use Slido. So open up that Slido link that you had. Oops. Open up that Slido link that you had. And the first question is for the quiz. We're not going to use your name. We're going to anonymize it. So you don't have to be embarrassed about any of your answers. And to make your name, I want you to use your favorite color and your favorite animal and put them together. So for example, like blue lion or red sloth. So enter your name into Slido so I can see that you are indeed in Slido at the moment. Brilliant. Okay, so we've got Joe and Blue Panda. So it's going to be a competition between the two of you.
Okay.
So how it's going to work is I have a series of questions, and with these questions, we're going to try and explore the Ensembl Beta site together. So the first question is, what tools are currently available in Ensembl Beta? So what I want you to do is go to Ensembl Beta, find out what tools you can use, and select the tool or tools in Slido and submit your answer. Once you've done that, I'm gonna show you how you can find this in Ensembl Beta. So I want you to try it out first on your own, and then we're gonna do it together to see how you can find that information. I'll just give you a couple minutes, a couple moments for this. Okay, so how do you find that information? How can you find out what tools that are currently available in Ensembl Beta? So first thing you want to do is go to the home page, and if you paid attention in the presentation, you'll probably remember that the only tool that is available is BLAST. So up here, you can find it. All right, everyone's correct. Either you paid attention really well or you found it on your own. Either way, this is great. So next question. How many Triticum aestivum or bread wheat genome assemblies are currently available in Ensembl Beta? Is it 1, is it 17, or is it 18? So again, go to Ensembl Beta and find out how many bread wheat assemblies we've got. Okay, so we've got 18 people answered 18. So let's check if that's correct. Let's switch back to the Ensembl Beta browser. The first thing you need to do is go to the Species Selector app. And you can either search for Triticum aestivum using this text box, or you can just scroll down to the species list. And just out of memory, I know this is bread wheat. And indeed, there are 18 assemblies available for bread wheat. Amazing. All of you got it correct. So next, how many coding genes does the human GRCH38 genome assembly have? Is it 20,787, 20,481, or 19,950? So again, I'll give you a few moments to find this out. Oh no, sorry. I didn't realize that that happens. Okay, here we go. All right, let's see. People are saying 20,481. So let's check if that's right. Back to the Ensembl beta page. We already had the GRCH38 assembly selected from the demonstration. But if you haven't yet, you can search for human in the text box, click on find. In the table underneath, there the first entry is the GRCH38. So you'll need to select to add and then add the species to see it up here. In my case, I've already added it, so I'm going to close this panel. And then if you want assembly information, such as the number of coding genes, you just need to select or click on the assembly. And from this panel, we can see there are 20,481 coding genes. And indeed, that's correct. Amazing. Cool. So next question. When was the gene annotation of the human GRCH38 genome last updated? Was it December 2013, January 22, or September 23? I'll be honest, this one was a bit more tricky. People are saying September 23. Okay, let's check on the browser. So I'm going to go back to Ensembl Beta. And for any annotation information, you want to be on this track panel on the right. Under annotation, you can find when it was last updated. So September 2023. And you got it right. So it wasn't that hard to find after all. Amazing. Next question. What Ensembl app do I use to visualize the genomic region? You might not need to check on Ensembl Beta for this. So if you remember, just select your answer. If not, feel free to find it on the browser. All right, very easy. I should have left this question out, but you use the genome browser to visualize genomic regions. You can visualize the genomic feature using the entity viewer as well. But if you want to explore any neighboring genes or neighboring genetic features, you will need to use the Genome Browser app. Okay, so next I want you to search for the MYC gene in human GRCH38. And I want you to tell me how many transcripts the MYC gene has. Is it 1? Is it 14? Or is it 15? All right, let's see. Some people are saying 14, some say 15, so let's check. Let's switch to our browser and search for the MYC gene. So to do so, I need to close this assembly panel first. Click on Find Gene at the top of the page and enter MYC into the text box and click Go. So because I've still got all the three human genome assemblies selected up here, I'm getting results for all of them, but I'm only interested in GRCH38, so that's the first one, and it's the MYC gene that I'm interested in. So I'm going to click on this, and you can either open it in the genome browser or in the entity viewer. I'm going to open it in the genome browser because you'll be able to see the number of transcripts in both of them. So here we have our MYC gene highlighted in the very first track. So this transcript, this is one transcript, and then it says plus 14. So it's actually 15. So 1 plus 14, all the other transcripts. Or you can also, if you go to the track panel on the right, under genomic, if you expand all of the transcripts for MYC, you can count the individual transcripts, and you'll notice that these are 15 transcripts. So well done on those who answered 15. All right, so next question. What chromosome is the MYC gene located on? Is it 8, 12, or 15? Okay, people are saying 8, so let's check in Ensembl Beta. So let's, hang on. Let me close this panel just to make it a bit easier for me. Several things you can do in here. Currently, I'm zoomed in. So if you're zoomed out, you should be able to see the coordinates where my mouse is right now. So 8. Sorry, the coordinates are follow a very particular format. It starts with the chromosome number, followed by a colon, followed by the start position, hyphen, end position. So the first number here represents the chromosome. Or you can click on one of the MYC transcripts. And in the pop-up menu, you can find the coordinates in there as well. So again, we've got the chromosome, start position, end position. And indeed, it's located on chromosome 8. Huh? 12, okay. No, I think that was a mistake on my end. I didn't select the correct answer in Slido. So it is 8, don't worry.
Cool.
Okay, so stay in the genome browser. What is the closest protein coding gene on the reverse strand? Is it PVT1, LRAT-D2, or POU5F1B? Sorry. I have to say this one's a bit more tricky. Okay, LRAT-D2. So let's check on the genome browser. Um, First of all, let me close this pop-up menu and make or hide all of the other transcripts in NYC. So I mentioned in the during the demonstration, if you look at the background for the track, you can see in which direction which strand this is located on. So the NYC gene is located on the forward strand, but if you scroll down down the genome browser, you can find genes located on the, or you can find a track of genes located on the reverse strand. So what you need to do is zoom out and find until you find a gene on the reverse strand. So this might be a bit tricky if you have quite a small screen, but if you just keep scrolling out, let me just zoom out a little bit on my screen here. Okay, so if it freezes like it did for me here, just refresh the page and you should be able to find your your page again. So let's zoom out. Okay, so anything that's dark shade, that has a dark shade like this, are genes, protein coding genes, sorry. And anything that's a light shade like this, these are non-coding genes. So I asked for a gene, a protein coding gene on the reverse strand. So while the closest gene is the POU5 gene, it is located on the forward strand. And the closest gene on the reverse strand is indeed, if you look down here, LRAT-D2. So well done on those who got that one right. If you have any questions or if you want me to walk through anything again, please let me know in the chat and I'm happy to do so. Okay, next question. Open the MYC gene in the entity viewer. What's the corresponding RefSeq ID for the main select transcript? All right, let's see. People are saying the first ID. I'm not even going to read that out loud. So let's see if this is correct. Let's go to Ensembl Beta. I'm going to go back to my gene of interest, which is MYC. And to open the entity viewer, there are two options. You can either click on the transcript and open it in the pop-up menu, or you should also be able to find it in the track panel. So if you click on the MYC more information icon, there is a link to the entity viewer for that as well. So just making sure we're indeed in the entity viewer, we can check the icon in the top of the page. And we want to find out the RefSeq ID for the main select transcript. So in this case, this is highlighted here as the first transcript. and you can open or expand the more information panel and find some basic information, such as the matching RefSeq ID, which is NM ending in 7.6. Alternatively, you can also go to the track panel on the right, go to external references, and open the main select transcript, which is this first one, and find the ID in here. So 2 options you can do. Okay, so that's correct. Well done. Next question. Can you find a hit in the T2T genome that matches the protein sequences completely? If so, what is the matching protein ID? So this one's a bit tricky. Let me just show you how you can do a quick blast search, because I didn't show you during my demonstration. So what we're interested in is the main select transcript, and we want to perform a blast search of the sequence, a protein search of the sequence on the T2T genome. So if we go back to the genome browser, let's close this. If we go back to the genome browser for the MYC gene, we want to go to, you can go to the track panel on the right, click on more information on the main select transcript, and open the sequences panel down here or up here, and select the protein sequence. So once you've selected the protein sequence, you can, you don't have to copy this. You can just click on this blast icon, and that will send you straight to the blast tool with the sequence in there. So what you'll need to do next is select the species that you want to perform your search in. You can select multiple species, but in our case, it's just the T2T genome, and then just click run and see how many hits it's got. So hopefully that will help a little bit. So we've got 2 answers. The first ID ending in 4.1, and the second answer is there are no hits. So let's see how to do this on the browser. Let's switch to the Ensembl Beta browser. I've already selected my protein, or I've already opened the BLAST, the tool with the protein sequence. And what I need to do next is add my species of interest. So select species. At the moment, I don't have the T2T in my list. So what you need to do is click on the T2T assembly up here, or if you don't have it in your species list, you can just go back to the species selector and add a species using this plus button. I don't want to blast against GRCH38, so I'm going to delete that species. And all you need to do is click on run. So you'll know that you have a BLAST job running when you see this big red button next to the BLAST icon. At the moment it's running, and once it's finished, this results button will flash up green or blue. Sorry, green I had from the old one. So this one is now available. The results are now available to view. So let's open the results. Okay, so there are 29 hits in T2T. If you want to see the individual the individual hits, you can click on the T2T ID. This gives you a list. Oh, sorry. This gives you a list of all of the BLAST hits that you've got. And the question in the poll was, let me remind myself, are there any protein sequences, are there any matching protein IDs in T2T that matches the protein sequence completely? So what we want to focus on is the percent ID. Again, if you didn't hear it during the introduction, whenever you see this question mark here, you can always hover over it with your mouse to give you a description of it. So the percent ID will tell you how many residues have aligned with your reference sequence. So the MYC and GRCH38.
Luis, I think you are muted again. Sorry.
Sorry. Oh man, sorry about that. I had a little pop-up window there. Sorry. Okay. So whenever you see a question mark like this, you can hover over it with your mouse to give you a description. So the percent ID gives you the percent of how many residues align perfectly with your reference sequence. So in our case, the MYC GRCH38 one. So what we want to do is sort this table by percent ID. So you can just click on the column, and we can see this one has 100%, but we also need to check the length. So does it match the entire sequence? And from memory, the length of the protein is 454. So this very first one is the ID ending in 4.1. And I think that's one of the answers that you guys chose. Indeed. There we go. Well done. That was a tricky one. Okay. Final question, or second last. Retrieve the protein sequence. Oh, okay. This is the same question as the previous one, but better phrase. So I think what I meant to do there was delete this and keep this one. Sorry about that. Okay, So then we've got, in which species can you find the closest gene match in terms of protein similarity in percent? So what you need to do is look for the main select transcript in the entity viewer, so still MYC, and find out the closest match in the gene relationships tab. So let me just show you on my screen. I'm going to switch to the entity viewer, and I want to make sure that I'm still in the MYC gene. So you can see that in this gray navigation bar up here. What you can do is scroll down on the left-hand side, go to gene relationships, and these are this is a list of all of the homologs found for MYC. So you want to find out the closest match in terms of protein similarity. Okay, so let's see what people have answered. We've got domestic pig, domestic guinea pig, and we've got chimp. So let's check on the beta browser. In gene relationships, we can sort the table by protein similarity by clicking on the header. So sometimes you need to click it twice just to see if it's in ascending or descending order. So I want it in descending order. We've got 91% domestic pig. Oh, sorry, hang on. Oh, there we go. Sorry. So let's start from the beginning. So this is your default view. If you click the first time, it will sort it in descending order. And then if you click it another time, it's in ascending order. And then it goes back to the default view. So if you click it once, we've got 99% protein similarity for chimp. And indeed, that's what most of you have answered. Well done. All right. So good job, Green Moth. You've answered the most number of quiz questions correctly. Well done. I don't have a prize for you. I'm very sorry, but you can call yourself a semi-expert in Ensembl Beta now. So well done. Okay, so we are a bit over time. Don't worry, I will still give you half an hour of break. So we're gonna do that now. Please be back. What time is it now? Let me just check. Yeah, please be back at 12:10, so 10 past 12 Colombian time. Please do get up from your computers and get yourself something to drink. There is plenty more practicals coming up after the break. So I'll turn off my camera for half an hour and I'll be back here. I'll leave the chat open. So if you have any questions in the meantime, please feel free to add them to the Q&A or into the living document. Thanks. See you in half an hour. Okay, welcome back, everyone. Hope you had a nice break. I thought my coffee would last longer, but I already finished it. Hope you got something to drink next to you there. So for the rest of the session, we are going to do a practical. So these are exercises in the training page, and I will share the URL with you again, just in case you missed it. But before we move on to those practicals, I just want to talk about genes and transcripts in Ensembl and how we annotate these. So while we are going to have a new Ensembl Genome Browser, the underlying data is still the same. So all the processes, all the the ways that the genomic data is being handled by our teams remains the same. It's just the interface that's changing, really. So I think that talking about how we annotate genes and transcripts will be quite useful in case you weren't aware of this, of how Ensembl does this. Okay, so let's talk about how we actually annotate genes in Ensembl. Genomes that are annotated with genes are referred to as gene builds, and this is a term that you will come across quite a lot when you work with genome browsers. So gene builds are just genomes with annotated genes. There are two independent annotation methods that we use in Ensembl. So we've got the Ensembl automatic annotation pipeline and the Havana manual annotation pipeline. But both of these annotation methods rely on valid biological experimental data. So that means that we only add gene annotation when there is experimental evidence that a gene is transcribed. The automatic annotation, Ensembl automatic annotation method, is applied to all vertebrate genomes in Ensembl, whereas the manual annotation method is only applied to a handful of species. This is because the manual annotation is done by real people, and it takes much longer to annotate genes this way. So the manual annotation is done for human, for mouse, rat, pig, and other agriculturally important animals. So partially cow as well, and a few fish species. The automatic annotation method is a gene prediction pipeline, but it's not just looking at open reading frames, but rather what the teams do is they take known data. So for example, transcripts, proteins, cDNA, and RNA-seq data, and they map them to the genome. And it can take up to 2 weeks to annotate a single genome using the automatic annotation pipeline. This is just a list of the annotation sources that we use in the Ensembl automatic annotation pipeline. So we retrieve nucleotide data such as cDNA and RNA-seq data that's submitted to the International Nucleotide Sequence Database Collaboration, or INSDC. This is made up of the NCBI, DDBJ, which is the database of Japan, and the European Nucleotide Archive. We also retrieve proteomics data from UniProt, and if there is not enough experimental data, so for example, for a lesser studied organism, we also perform annotations based on gene prediction by sequence similarity of a closely related organism. So for example, human and chimp. The manual annotation is done by the Havana team. So Havana stands for Human and Vertebrate Analysis and Annotation. So to begin with, manual curation was only done on the human and mouse genomes. This makes up the GenCode gene set. But since then, we've added, or the Havana team have added more genomes to their list. The Havana team curates annotation for zebrafish and pig in the whole genome. They also curate annotation for specific regions in rat, hamster, and cow. And we collaborate with research communities to annotate other well-studied species like chicken, tropical clod frog, I think this is, and bread wheat. In the manual annotation pipeline, the annotation is done by a person on a case-by-case basis. So what they do is they look at specific data sets such as RNA-seq and transcript structure data that has been published. So it has to be publicly available because all of the data in Ensembl is public. We want to make sure everyone has equal access to them. But as a manual curator, they have a wider range of evidence that they can retrieve data from, because there's more flexibility compared to the automatic pipeline that runs through, through a computer. It has to be very specific data sets. Whereas in the Havana manual annotation, they can look at very, what's the word? Very, very wider range of data. Sorry, a wider range of data formats. There we go. It doesn't have to be a very specific. Oh, and I forgot to mention, the Havana manual annotation is also much more laborious, and it takes more time. So it can take up to half a day to annotate a single gene, and to annotate a single genome, it takes several years. So this is another reason why we only do it for specific, or for a handful of species in Ensembl. Comparing the manual versus automatic gene annotation, the manual annotation is much more comprehensive, and it is more accurate for difficult features such as splice sites, but it also takes much more time. And if we had the resources, if we had the opportunity to hire as many manual curators as we could, I think we definitely would, but the funding is not there. And this is one of the reasons why we only do it for a handful of species. So I briefly mentioned the GenCode gene set. So this refers to the human and mouse gene builds. So the GenCode project focuses on these two species, and it's the merged gene set of the Ensembl automatically annotated and the Havana manually curated genes. And it's also the default gene set used by many major projects like NOMAD, the 100,000 Genomes project by Genomics England, ENCODE, Blueprint, and so on. Another project to know about as well is the main select. So main select dictates the annotation confidence in a transcript, and main stands for matched annotation between NCVI and Ensembl EBI. So NCVI have their own annotation pipeline, and we have our own annotation pipeline. And if both of these pipelines, both of these independent pipelines, annotate a transcript the same way, they are flagged as main select transcripts. So it just tells you that there is more confidence in the annotation of that transcript. Main select transcripts are 100% identical between NCVI and Ensembl EBI. And it's also agreed to be the most biologically relevant and the representative transcript of a gene. So if you are if you're not sure which transcript to take to further study, the main select transcript is a good start in this case. And then elaborating on transcripts. Sorry, I forgot to mention, for main select, this only applies for human. For any other species, we use the flag Ensembl canonical transcript. So for species where you have genes with multiple transcripts, we select one transcript to be the representative transcript. And this is usually the most highly conserved, the most highly expressed. It's also in concordant with other transcript scoring algorithms, and they have the longest coding sequence and the largest number of clinically relevant variants. And wherever available in human, the Ensembl canonical transcript is the main select transcript. So all features in Ensembl have an assigned Ensembl stable ID. Whenever you see an ID that starts with ENS, you can be sure that this was annotated by Ensembl. So our IDs start with ENS, followed by a single letter, which represents the feature type. So for example, G for gene, T for transcript, and so on. And then this is followed by an 11-digit number and a version number after a dot. So if an annotation slightly changes between releases, we increment the version number. This is the case for human. For any non-human species, we also add a suffix after the ENS. So for example, for mouse, we add MUS after ENS, for zebrafish, DAR, and so on. And you can find a list of all these suffixes in the link at the bottom of my slide here. Now, if you're interested in the more technical bits of the gene annotation pipeline in Ensembl, there is a very interesting paper from our colleagues, the Ensembl gene annotation system. And there's also more information about the GenCode gene sets. All right, so let's move on to a demonstration. We're going to use Ensembl Beta for this. And what we're going to do is we're going to look at the human UQ-CRQ gene and find out what information we can find about the gene and its transcripts. So let's switch to the browser. And I'm actually going to start right here. So you can start on the home page if you like, but I want to show you different ways of searching the new site. So in the entity viewer, if you go to the track panel on the right, you can click on the search icon and you can enter your gene of interest in here. So for us, it's UQ-CRQ. And I just want to make sure that I'm in the right assembly as well. So I'm interested in the GRCH38 assembly, and indeed, this is highlighted up here. Okay, so to start your search, click on Go. And there's only one hit for UQ-CRQ. So if we click on the ID, we can find more information about it. Oh, let me just refresh the page because the icons aren't loading. So search UQ-CRQ and then Go. And if you click the ID, it will tell you more information about the gene, and you can view the gene in the genome browser or in the entity viewer. I'm going to look at it in the genome browser first. So we're quite familiar with this already, but just to recap, our gene of interest is always going to be on the first track, so UQ-CRQ. The one that's being displayed is always going to be the main select or the canonical transcript, so main select for human. And if main select isn't available, it's the canonical transcript. You can expand your gene to view all the other transcripts as well by clicking on this plus icon underneath. If you want to find more information about the individual transcripts, you can either click on the transcript in the genome browser itself, or if you go to the track panel on the right, you can expand the list of transcripts and click on more information. And this gives you even more information than the pop-up window. And here's where you can view the sequence as well, or download the sequence to your machine. Let's close this panel. So back in the genome browser, there are variation and regulation tracks available. To find them, you just need to go down in the genome browser. And hopefully you can see this, but we've got the regulation track first. So this is one regulatory feature, and then we've got the variation track. And the different colors represent different types of features. So if we want to see what the red in the regulatory track means, we can switch to regulation in the track panel. And scrolling down, you can see the different colors. So red stands for promoter. You can also click on this. That will also tell you what type of regulatory feature this is. Okay, let's close that. If you want to, if you want to look at the individual variants, you will need to scroll in, because a lot of the variants, it just looks like one big variant, like this one here, but it's actually a lot of smaller variants. So if you want to look at a variant, you might need to scroll in a lot. And again, you can switch to the variation tab on the right to see what type of variants you've got in your sequence. So let's just look for let's click on one of these variants. Let's just take this variant here. So if you click on the variant, if the variant has an ID, we will provide that for you. We also give you the most severe consequence. So this is the sequence ontology term. So the standardized vocabulary for variant consequences. We give you the type of variant. So this is a single nucleotide variant, SNV, the location, and then the alleles. So the first one is always the reference allele, and the second one is the alternative allele. You can view this in the entity viewer if you like. And in the entity viewer, you'll find from which database we import the variant from. So all the variants that you can find in Ensembl, they are imported from publicly available archives, but we apply our own analyses to them. So what we do is we map each of the variants that we import to the genome. And depending on where it falls within a gene, we apply a consequence to it. And that's this sequence ontology term that you're seeing here on the right. So the one I've just clicked on is the variant ending in 8486 SNV. We already saw this in the genome browser. The reference allele is T, the alternative allele is C, and this is these are the coordinates for the variant. In the track panel on the right, we can see that the variant was imported from dbSNP release 156. Again, we've got our alleles, we've got our location. We also have our variant in variant call format, VCF. And from here, you can also go back to your genome browser and explore any neighboring variants that way. One thing I actually didn't mention earlier were these icons here in the panel, in the track panel. So you can make this track panel a bit smaller if you like. You can hide it. There is a search option here as well. So this is where you can enter a gene. This one here will give you the history, your history, so all the genes that you've searched for. This is not yet available, but this will be a way to generate a URL to share the page with your colleagues. And here you can download the sequence. In this case, this is not yet available for the variant. All right, so I think this is all I've wanted to show you for this demonstration. We now have time for exercises. We have quite a lot of time for exercises, actually. And this is going to be, it's not going to be interactive this time, I'm very sorry. But the exercises you can find in the training page. So this is the training page. And if you scroll down to the bottom, open up the Exploring Gene Builds in Ensembl Beta. And we've got several exercises here. So if you're studying human, start off with the human exercises. There's also one bacterium exercise and a wheat exercise if you're finished with the human ones. And this is just to make you become more familiar with the Ensembl beta site. So it's ways of accessing the data in the browser. If you need any help, we are here in the background, so please feel free to ask away in the Q&A or in the living document. Otherwise, there's also text answers here. So if you need a little nudge, a little reminder of how to get to the data, you can open up the text answer. And then final thing, this training page will be available to you indefinitely. So just like the living document, as long as there's internet, you will have access to this training page. So if you don't have time to finish all the exercises, don't worry, you can always come back to it in 2 weeks' time and try them out. So we'll be here in the background. If you need any help at all, please don't be shy. And I'll check back with you in maybe half an hour's time to see how it's going.
And I'll be on the call, anyone of you, if you have any questions while you're working, we'll go through it together.
If you'd like to go through any of the exercises together, just let us know and we're happy to do that with you.
I mean, Louise, you can have a break. That'll be Juan and I tackling this.
Oh, okay. That's even better. Yeah, I might just grab another coffee if that's okay.
Go for that. Go.
All right, thank you.
As I play with the browser, is if you shrink or expand your screen, like stuff hides, like depending on how many stretching menus you have. So I've just noticed that sometimes I'm like, wait a minute, where's this thing that I just saw? No, it's a little bit smaller, so it completely hides.
That's good to know.
A bug? I don't know. Let's see if this happens for you guys too. What happens if you search for this whole string, for like the name of Escherichia coli, with its brackets in the search box for species selected?
All right, let me find out.
Yeah.
Ha! Yeah, that looks like a bug.
Bug found.
Does it work without it though?
It does.
Okay.
It does. Yeah. So I don't know if brackets induce the fail.
Yep.
Bracket. Like, let's just put a bracket for something. A bracket causes search to fail.
Yep.
Just
That's good to know.
do other things cause it to fail?
Is it?
Let me try a star and add thing. Yeah.
Any symbol?
An ampersand. Wait, a star. No, star fine. Ampersand. No, that's fine. Dollar sign. Maybe double on if you double them up. Okay, cool. We have a bug to report.
There we go.
Cool.
And if you guys, you participants, see anything like this, let us know. Very good question. So Joanna asked, how do we report a bug? I think the easiest or the most straightforward way is if you go to the Ensembl beta site, we can click on this form, on this contact us form in the top right, and just enter your bug in here. It usually helps if you include a screenshot. That'll help us find the bug much quicker. Alternatively, you can also email us, and I will leave the email address in the chat. No worries. Okay, I take that as finished. If not, let me know. Last chance. Okay. In that case, I think I'm happy to move on. If you didn't quite have enough time for the exercises, just know they will be available to you after the workshop as well. So you can always come back to them. So let's wrap up our tutorial for today. And to begin, I just want to make you aware of leaving some feedback for the Ensembl beta browser. So if you were able to explore the browser a little bit and you had some thoughts about maybe the interface or any functions that that you notice we could have or we don't really need, you can leave a feedback, or you can leave, feedback for the browser using this button in the very bottom left of the homepage. This will open a little survey, and it'll just ask you a bunch of questions of how did you find it, what did you like less about the new browser, and so on. Alternatively, you can also leave a comment using the contact form in the top right. So that will be sent straight to our help desk. Right. So for the workshop, we do have a separate feedback survey. To find the link for the survey, you can go to the training page, and there is a link just above the exercises. I will share that link in the chat as well. So if you open the link, there are a few questions. It shouldn't take any more than 3 minutes, but there are a few questions about your experience of the tutorial. And if you like, you can also retrieve a certificate of attendance. So towards the very end of the survey, there is a question that asks you, do you want to get a certificate? Just click on yes. And in the next page, just enter your email address in there, and it will be sent to your inbox straight away. So I'll give you a few minutes for this now, and then we'll just summarize anything we learned today. And of course, we'll be around for final questions towards the end as well. All right, while you're finishing up the feedback, let me just go through some closing slides. Oh, there we go. So what did we learn today? We learned all about the new Ensembl browser. So Ensembl is a genome browser that integrates genomic data. It allows you to search and retrieve and analyze the different genomic features in a particular species. We've got annotation for genes, variants, regulatory regions. We've got comparative genomics analyses. And we also have a number of different tools that you can use to analyze your own data. The Ensembl beta browser is going to be the future of Ensembl. So please keep in mind that this is still in the beta phase. So if you have any feedback at all, now is the time to tell us, because now we still have a chance to change that before the final release comes out. Ensembl data is completely open source, just something to keep in mind. So you can use the data as you wish in your research, and completely free of charge. And what I think Ensembl is really powerful in is it's an added value resource. So what it does is it brings together information from a wide range of other databases into a single site. So it's kind of like a Google for genomes, basically, but less finances, just like anything in science, really. So if you use Ensembl data, we do ask you to cite the most recent paper. And I have a slide for that most recent paper towards the end of the presentation. Just a quick question from Joanna. Are there any plans to add more genomes of other species like bacteria? Yes, yeah, so for sure. There is a separate portal for bacterial genomes, and it is called bacteria. You can access it via bacteria.ensembl.org. So that is the current Ensembl site for bacteria. But with the new site, we're actually going to add all of the genomes into a single platform. So instead of accessing all the data using different portals, in future, you'll be able to access it all from the new site. Very good question. Thank you. So just a summary of the different sites that are currently available. If you want to access any vertebrate genomes, you can go to the main website. For any non-vertebrates, you'll have to use the Ensembl Genome's sister site. We also have the Ensembl Rapid Release website, and I think I didn't really talk about this, but this portal is updated was updated every 2 weeks. And the sole purpose for that platform was to quickly provide annotation for genomes that have been sequenced recently. So from projects like the Darwin Tree of Life, which aims to sequence all of the different organisms across the UK and Ireland. We also have a portal for the GRCH37 human assembly, the older one, and one for the coronavirus genome. While we work in release cycles, we do keep all of the archives of previous releases. So if you need to access any old releases, you can either look for it on the Ensembl website, or you can access them you can access the data via our FTP site as well. And the Ensembl beta site is the one that we explore today. So this is going to be the future of Ensembl, very fast and modern looking. If you want to access data programmatically, current data, the current Ensembl data, you can do so via our FTP site. We also have a REST API, Perl API, and we have MySQL servers as well. Now, this was just a very small fraction of the usual courses that we Ensembl Outreach provide. We do provide full-day courses on the Ensembl browser. So if you would like to organize something like this at your institute, please do get in touch with us. We can do a browser course on the current browser or the beta browser. We also have more advanced courses, one that is focusing on how to access Ensembl data via the REST API. And we also have a train the trainer course where we teach you how to teach Ensembl. If you are interested, please leave us an email at helpdesk@ensembl.org. You can also use this email for any feedback or any comments or any Ensembl related questions. And if you just want to learn more information about hosting an Ensembl workshop, you can go to the training page for that. If you want to reach out to us on social media, we are on Facebook, we're on LinkedIn and X. And for any release updates, for example, or job opportunities, you can go to our blog, ensembl.info, for that. A few helpful links. So the EBI, the European Bioinformatics Institute, has a very good training portal. So you can find anything bioinformatics related training on this website. If you are quite new to bioinformatics, you can also find introductory courses in there. There's one that's called Bioinformatics for the Terrified, which is a really good overview of the different resources that you would use in computational biology. We also have Ensembl focused tutorials on our website and our YouTube channel. And again, if you want to reach out to us, you can email us at helpdesk@ensembl.org, or you can sign up to our mailing lists if you want to get any updates directly to your email. Now, if you're using Ensembl data, please do cite the most recent paper. This would be Ensembl 2024 for the vertebrate species. And if you're using non-vertebrate species, gene annotation from non-vertebrate species, you'll need to cite this second paper, Ensembl genomes, as well. All right, so it's not just Juan, George, and me in the team, of course. We're actually a team of about 100 people now, so it takes a lot of different backgrounds to keep a resource like Ensembl going. So I just want to thank all of them and all of our funders as well, and thank you, participants, for staying until the very end. I believe the whole tutorial is recorded, so if you've missed anything at all, please come back to the recording. If you have any questions at all, we will leave the room open for, let's say, another 10 to 15 minutes. And yeah, thank you so much for your attention and the amazing quiz.
Just like to thank you, Luis, for taking us through today so excitingly and carefully through a new browser, which is set to be the next 20 years or so of browsing genomics on Ensembl. So it's a really exciting point to be in the future of genomics. And thank you for taking us through and with some great details. So I really enjoyed being here.
Thank you.
Thank you to our attendees, and if you're watching the recording, thank you very much, and we hope that you've enjoyed what you've learned. And if you have any feedback for us, please use that help desk email and get in touch. Before we end?
Hope that you enjoy, as I did, attending this course as well, because I learned so much from you, Luis. So thank you very much.
I also want to say for the participants who joined us, this is our first beta focused workshop, or kind of longer workshop. So you guys are pioneers as well. So thanks for watching the recording and being here in the room.